So you have learned about the biosynthesis of cholesterol. Now let's talk about naturally the degradation of this compound. In contrast to other compounds like glucose or like proteins or sort of lipids like fatty acids, the cholesterol is not degraded into carbon dioxide and water because it's not mainly an energy producing compound. Its main function lies in its very structure. So, though it's not degraded into carbon dioxide and water, but some part of it or some of the cholesterol has to be excreted, and it is excreted through the bile. You know, bile is one of the very major product of cholesterol. The cholesterol is secreted into the bile, and then bile transport the cholesterol from liver towards the intestine. In the intestine, there is further modification which takes place to cholesterol by the activity of bacteria. For instance, you get the secondary cholesterol types like coprostanol and cholestanol. Both of these are excreted through the feces, and they act or consider an indicator of the presence of cholesterol in an organism. So environmental sciences, they are used as the indicators. As far as the composition of bile is concerned, it's a watery mixture, liquidy mixture of organic and inorganic compounds. Of course, you know that major portion, of course, will be the bile acid, which will be coming from the degradation of the cholesterol. In addition, it also contains the phosphatidylcholine, conjugated salts. Both of these are key components or key organic components of the bile. It passes from liver towards the excretory through in its excretory pathway. It passes from the liver to the duodenum, from where it's excreted. It is stored in the gallbladder. It's this the bile is stored mainly in the gallbladder. That's why the gallbladder is the storing organ of the bile. The structure of the bile is, because it comes from the degradation of cholesterol, is a derivative of cholesterol, you can say. So that's why it's very much similar in terms of structure to its parent molecule, that is the cholesterol. It contains 24 carbons, have 2 to 3 hydroxyl groups. Its side chains have a carboxyl group, and you can see it very clearly. It forms bile acids after degradation. The cholesterol after degradation gives rise to bile in the form of bile acid. In duodenum, it is half protonated, and in the form of bile salts, it is half protonated and half deprotonated. Occurs in two forms in the large intestine. One of the major function of bile, and you know it, we have talked a lot about it, it acts as an emulsifying agent which contributes towards the digestion of the lipids. As far as the synthesis of bile is concerned, it's a very multi-step complex process. It involves the reduction of the cholesterol double bonds, shortening of the hydrocarbon chains, and introduction of the carboxyl group. You can see it here that this is the main cholesterol compound, and it gives rise to the formation of the primary bile acid, that is 7-alpha hydroxycholesterol, and it is a reducing event because it's a reducing reaction because it requires energy. It's a biosynthesis. So, biosynthetic reactions involve the investment of energy. Then 7-alpha hydroxycholesterol gives rise to two types of bile acids. One is the cholic acid, which is a triol, means it has 3 hydroxyl group, and one is the diol, which means it has 2 hydroxy groups. This is a rate limiting step also from cholesterol to 7 alpha hydroxy cholesterol, because almost all the pathways which involve the biosynthesis of products, biosynthesis of different compounds, they regulated by feedback mechanisms. Here again, the negative feedback mechanisms regulate the level of production of bile. So more bile acid will definitely negatively regulate this reaction. One thing I must tell you, and I have already told you, that it's not as much important to know that what are actually the products and the reactants of reaction. What you should know is that why these products are being formed and under what circumstances they are being formed. What is the environment in which this is being formed? The bile here is being formed in an environment which have it's occurring, this formation is occurring in the liver. So why bile has to be formed? For two reasons. Bile has to be formed for the excretion of the cholesterol also, and also it has to be formed for the emulsification process. So these are the things you should always keep in mind whenever you are studying different chemical pathways. It's not always about the reactant and products which are involved. It is also about the regulation and the environment in which this whole process is being regulated.